Good afternoon, children. Last class we have discussed principles of inheritance. These neat questions. What the questions we discussed here, you do the practice. After completion of practice, any doubts are there, seek clarification through WhatsApp. Relevant with the question and answers. While solving the paper, any doubts are there, seek clarifications. Then the remaining questions we have to see. Today and tomorrow we will conclude these questions, principles of inheritance. Later we will start the next chapter, molecular basis. Then the question number now, 31 question. You see already you are served with the sheets. Once you check which one of the following symbol and its representation used in pedigree analysis is correct. You have to choose the correct symbol. You see here, question number 31. This is the principles of inheritance. Principles of inheritance and variations. Principles of inheritance and variations. Question number 31. Here. Which one of the following symbols? Which one of the following symbols and its representation used in human pedigree analysis. Used in human pedigree analysis is that what is the pedigree analysis is correct. This is A, option A. Mating between relatives. Mating between relatives. Unaffected male. This is unaffected male. Unaffected female. Unaffected female. Male affected. Male affected. Already you know this here pedigree analysis chart. In this pedigree analysis chart, what symbols we are using? If it is circle, circle is there. This is the circle symbol. Circle for male or female. This is circle for female. Female is there. But this is a circle. It is also circle. Difference is what is here. This is darkened circle. Here circle without darken, normal female. This female is called normal female. The same circle it has been darkened. How it will be called darkened circle? Affected female. Darkened circle means here affected female. Circle is darkened, affected female. Next one here you have what we can use here, square. What is the symbol of square? This square without darken, how it will be representing normal male. This is the normal male. If this square, same square, this is darkened. How this is called darkened square? This is the affected male. This is affected male. Square is darkened, affected male. Square is without darkened, normal male. This is the what is here? Male. And now you see this another symbol. What is here? This is a rhombus symbol. It will be there. How it will be there? Sex unspecified. Sex unspecified. That means not male, not female. This is not affected, normal. But here, what is the case here? Normal sex unspecified. Same thing is darkened. Suppose here, like this is there, here it would be darkened. How it will be there? Here, affected sex unspecified. Affected sex unspecified. This is the unspecified sex is here affected. Next symbols. What is the symbols here? Mating. What is the mating? Mating is nothing between male and female. This mating, how it will be called? Normal mating. If one line is there between male and female, this is a female, this is a male. Between male and female, one line is there. How it will be called here? Normal mating. This is normal mating here. That means normal mating means here mating happen between anyone. But here mating here between two lines are there. How it will be called here? This mating is called as this here consanguineous mating. That means mating between relatives. If a marriage is held between those relatives, then it will be called consanguineous or mating between relatives. This symbol, how it will be called mating between relatives. This is also called as the here consanguineous mating. Consanguineous, consanguineous mating. Consanguineous mating. Consanguineous mating is nothing but here mating between the relatives. So here close relatives mating is there, mating between relatives. It has been showing with the two lines between male and female. Here, what is the correct one? This is first one, mating between relatives, correct only. Unaffected only, not male, it is female. This is wrong. Here unaffected only, not female, it is male. This is wrong. Here, this is a not male and not affected. Both cases wrong. So that what is the answer? The option A, mating between relatives, that is the correct one. This is relevant with the pedigree chart. So the symbols, what are using in pedigree chart, you refer. Then this is relevant with the pedigree chart symbols. Next one, another question. Question number 32. ABO blood groups in humans are controlled by the gene I. ABO blood groups are controlled by what is the gene? I gene. It has three alleles, IA, IB, I0. Since there are three different alleles, 6 different genotypes are possible. How many phenotypes can occur? This is relevant with the blood group. What is the blood group here? Blood group gene. The question, what is the question? Blood group gene. Blood group gene, what is this blood group gene? I. I has how many alleles here? IG has 3 alleles. What are the 3 alleles? IA, IB, I0. 3 alleles. These 3 alleles, what can produce here? 3 alleles responsible for 3 alleles responsible for here. How many genotypes? 6 genotypes. These are the 6 genotypes. Then what would be the phenotypes? What would be the number of phenotypes? The question is, what is the question? You find out this year number of phenotypes. This question is relevant with ABO blood group. What is the question? Blood group G, I. I gene has 3 alleles. 3 alleles responsible for 6 genotypes. Then what are the number of phenotypes here? Options are given. Option A, 3 phenotypes. Option B, here 1 phenotype. Option C, 4 phenotypes. Option D, here 2 phenotypes. These are the number of phenotypes. Now you see blood group gene. This is the I gene. But I gene, how many alleles will be there? IA allele, IB allele, I0 allele. These 3 alleles. But how they have been arranged here? They have been arranged maybe combination IA with A, IA, IA. Next IA with I small I0. Or otherwise, what is this here? IA, IB. Like this here, combinations will be there. Next one, IB. IB, what is the combination? IB with IB. Next one, IB with I0. But here IV with IA already came, repetition. Next here I0, I0 combination. Yeah, I0 A already came. I0 B already came. Total how many combinations here? These are the 6 combinations. But what is this blood group? A group. What is this blood group? A group. What is this blood group? AB group. What is this blood group? B group. What is this blood group here? B group. What is this blood group here? O group. So these two comes under one phenotype. These two comes under one phenotype. This one comes under one phenotype. This one comes under one phenotype. Genotype 6. This is A group only, one phenotype. This is AB group only. This is another phenotype. These two B's comes under one phenotype, another phenotype. O group, one phenotype. Total how many phenotypes have come? 4 phenotypes. Blood group gene, I gene, has how many alleles here? 3 different alleles. 3 different alleles. How many genotypes they have been arranged? 6 genotypes. Alleles are 3, but genotypes are 6. Phenotypes are 4. So the answer, what is the answer? 4 phenotypes are possible. Answer is the C. So this is the 32 question. Next 30. Question number 33. You see, select the correct statement from the one given below with respect to diabetic cross. Tightly linked genes on same chromosome show higher recombination. Genes for apart on the same chromosome show very few recombination. Option C, genes loosely linked on the same chromosome show similar recombination. Tightly linked genes on same chromosome show very few recombinations. These 4 options here. This is the what is here. Select the correct statement. Select the correct statement. Correct statement regarding dihybrid cross here. Relevant with the dihybrid cross here. So select the correct statement. What is the first one here? Tightly linked genes. Option 1. Genes are tightly linked. Tightly linked genes. So what they are showing tightly linked genes? Here they are showing the higher recombination. Tightly linked genes on same chromosome. On same chromosome. So high recombination. High recombination. Option 1. Option genes apart on the same chromosome. Apart on the same chromosome. Genes apart on the same chromosome show very few recombinations. Very few recombinations. Option C, genes loosely linked on same chromosome. Genes loosely linked. Loosely linked on the same chromosome. The same chromosome. Same chromosome. Shows similar recombinations. Similar recombination. Option D. Yes, tightly linked genes on same chromosome. Tightly linked genes. Tightly linked genes on same chromosome. Tightly linked genes on same chromosome. So very few recombinations. So very few recombinations. Very few recombinations. You see in this case here, few recombinations means generally the given question relevant with the linkage and recombination. This concept is, this question is asked here, linkage. Linkage where it is depends distance between the genes. Linkage and recombination, these are the phenomena here. These two phenomena Linkage recombination, where it will be differentiate distance between the genes. Here, distance between genes affecting the linkage and phenomenon. Suppose what is this here? Distance increases. Or what about this here? Recombination increases. You see here phenomenon distance between genes is distance between genes. This is directly proportional to recombination. Increases the distance. What about the recombination? Increases. Distance between the genes would be increased, then recombination would be increased. Distance is decreased, then what about the linkage here? Increases. That means closely linked. Inverse proportional, direct proportional relation, recombination. Inverse proportional relation, linkage. Suppose here distance between the genes. Distance between the genes. What the relation will be there? Inverse proportional relation. Here I am writing. Distance between the genes. Distance between the genes inverse proportional to the linkage phenomenon. The phenomenon of linkage indirect proportional to the distance between genes. Distance increase, recombination increase. Distance increase, linkage decreases here. So what would be there here? Distance is playing a prominent role. Now you see here these are the genes. A gene, B gene. Now A and B. And you see A and B gene, in between what would be there? 5 organ units are there. Next, same genes. Here you see. Otherwise, here P gene, Q gene. Here between the genes here, 20 organ units distance is there. Which has this more recombination? P and Q. Why P and Q? More distance. Which have more linkage? A and B. Why more linkage in A and B? Distance is less. Then linkage is more. Tight linking genes are showing the more linkage. For distance arranged genes are showing more recombination. For distantly arranged genes showing more recombination. Tight linking genes are showing the more linkage. That is the conclusion. But from this conclusion, here tightly linked genes showing high recombination wrong. Tightly linked genes more linkage, not recombination. Genes apart of the same chromosome. That means genes are separated from same chromosome, but showing a very few recombination. If they are separated from same chromosome, going to another chromosome means more recombination, not few recombination. So this is wrong. More recombination. Here is high recombination is given. Wrong here. Low recombination. But genes loosely linked from same chromosome show similar recombination. Not similar recombination. Why? Because maybe genes, what would be there? A gene is there, B gene is there, different genes are there. Here A gene, B gene, C gene, and Q gene, something. But here this is loose packing is there, loose packing is there. But B and C, here how much recombination percentage? Suppose the distance between B and C 10 organ units, C and Q 20 organ units. But they are present on same chromosome. Here distance is far. It is high recombination. Compared with CQ, B and C less. So that here they are showing similar recombination wrong. Even though loose packing is there on same chromosome, but all are not showing the similar recombination. It decided by distance. If distance increase, recombination increase. So that they are not similar recombination. But tight linking genes on same chromosome showing few recombination. Recombination is less, linkage is more. If tight linking is there, what is more? Linkage. What is less? Recombination. So appropriate statement, what is this here? D is the correct. The meaning is here closely linked to genes showing more linkage, but recombination less. Therefore, the answer is the D answer is correct. So this is the D. The genotype of a plant showing the dominant phenotype can be determined by the genotype of a plant showing dominant. So to find out the genotype, what class has been done? Yesterday we discussed this is 34. Here the genotype of a plant showing dominant phenotype. If you know this here, genotype. This genotype, what it is showing here, phenotype is showing. Phenotype. Is genotype of a plant showing the phenotype. What type of phenotype? Dominant. But in this case, what cross we are using? Genotype means suppose A genotype is there. What is this here? Dominant phenotype. A is there. What is this here? Dominant phenotype. Small a is there. What type of phenotype here? Recessive phenotype. That means phenotype is decided by genotype. Genes are capital A. What phenotype express? Dominant phenotype. Capital A, dominant. Heterozygous dominant. Small a, recessive phenotype. So that is what character? Recessive character is expressing. Therefore, here what would be there here? According to genotype, what would be decided? Phenotype. Suppose A genes are responsible for tall. This plant is tall. This is also tall. Third one here, what is this? Dwarf. So this physical character, that means here appearance character or phenotype decided by genotype. But to find out genotype, what cross we are doing? Test cross. Yesterday we done test cross, dihybrid cross, pedigree analysis, back cross. To know the genotype of plant, what cross we are choosing? Test cross. So here, what is the answers here? These actions you see, one is test cross, A action, test cross, B action. What is B action? Dihybrid cross. B action, dihybrid cross. Action C, what is this here? Pedigree analysis. Pedigree analysis. Action D, what is the action D here? Back cross. So here, all back crosses are test cross. What would be their back cross here? Back cross may be dominant parent or recessive parent. All test crosses are back crosses, but all back crosses may not be test cross. But so that back cross means here, two actions will be there. Maybe dominant parent, recessive parent. So that we could not select this back cross. Compulsory, what we have to select here? Test cross. F1 with recessive parent, test cross. Helping for find out the genotype. Genotype is using for determination of phenotype. According to our gene configuration only, phenotype will come. Therefore, question is phenotype is decided by genotype. To find out genotype, what cross you are choosing? Answer is test cross. Back cross is not relevant. Dihybrid cross means different. It is for study of behavior of two pairs of contrast character, we are doing the dihybrid cross. And pedigree analysis, family history is studied by using various symbol pedigree analysis, not relevant to find out the genotype. Henceforth, what is the answer? Test cross is the answer. And now here you go for certifying Mendel law of dominance. Which one of the following is explained by Mendel law of dominance? Which one of the following cannot be explained on the basis of Mendel law of dominance? Okay. Which is not explained. Not explained by Mendel law of dominance. Law of dominance. Given question, which of the following statement is not explained by law of dominance? So here action A, which is not explained by Mendel law of dominance here. You see this here, discrete unit controlling a particular character called is factor. Here, discrete units controlling a particular character. Discrete units means genes. Discrete units. They can be called as factors. The discrete Mendel units, they are controlling particular character. They are called as a factor. First action. Second action, out of one pair of factor, one is dominant, other is recessive. Out of one pair, total one gene consists of how many here? One pair of alleles. Maybe capital A, small a. In these alleles, one is what is this here? Dominant. Another one is which will be called here recessive. Here, so what is the law of dominance? You see, there is a discrete units are there. These discrete units controlling particular character called as factor. Correct or wrong? This is so. This is correct only, but we cannot explain. Second one, the gene has two alleles. In these alleles here, one is dominant, one is recessive. Correct only. Option C. What is option C? Allele don't show any blending. Both the character recover as the gene F2 generation. Alleles don't show blending. Both character recover. Both character recover in F2. Alleles don't show blending. Both character recover in F2. Option C. Option D. Allele factors occurs in pairs. Factors occurs in pairs. You say this here, this is the law of dominance, this is the law of dominance, this is the law of dominance. But alleles don't show blending. But here both characters recover in F2 law of segregation. The alleles they cannot blend, but they are segregating into separate gametes. But both dominant parent character, recessive parent character recovered in F2 generation. That law which will be called law of segregation. Without blending, segregation possible. The segregated character recovered in F2 generation. What is this law? Law of segregation. But here you see what is the law of dominance here. The alleles are concerned. What would be their pairs? Out of pair, one is dominant, one is recessive. But this particular allele controlling the particular character. These three statements, A, B, these statements relevant with the dominance only. So that here C statement, what is the law of segregation? So what is the statement is not explaining the law of dominance here? Option C. So answer what is here? Option C is the correct answer. C is explaining what it is explaining here, law of segregation. So here this is the answer 35. Next one. This is a repeated question 36. I gene consists of the three alleles, don't show codominance. There are 6 genotypes. How many phenotypes in all are possible? 4 phenotypes. Question number 36. AB blood groups here. How many phenotypes? 4. Already this question one time came here. So 36 is what is here? AB blood groups. Here number of phenotypes here. What is here? Number of phenotypes in ABO blood groups. ABO blood groups. How many phenotypes are there in ABO blood groups? Number of phenotypes. Answer 4 phenotypes. The answer is the 4 phenotypes. Already this is it was repeated in a previous question. ABO blood group 6 genotypes are 4 phenotypes will be there. So number of phenotypes in ABO blood group 4. Pedigree chart certain family given below. Select correct conclusion which can be drawn from the character. You see this here according to pedigree chart here. Select the correct statement. You observe this pedigree chart. Affected male. He is a affected male and a normal female. Now they have a what would be the daughter, affected daughter, and affected daughter. Then here normal son. Then here like this it will be there. This is the here affected one. That means here what is the conclusion you have drawn? This is female. Female means what is here male. Male means the answers you see. Female parent is heterozygous. Female parent is heterozygous. Female parent is heterozygous. Next B. Parents could not have normal daughter for this character. Parents could not have normal daughter. Parents have had normal daughter. Could not have had normal daughter for this character. Next here, the trait under study could not be color blindness. trait under color blindness. The male parent is homozygous dominant. The trait is not colorblind. Is not colorblind. And the male parent homozygous dominant. The male parent. What is the male parent? Male parent homozygous dominant. Male parent is the homozygous dominant. These are the absence are given. Male parent is the homozygous dominant. Now you see what is the conclusion you can draw here. This is a XY. It is the XX. But here this is the problematic is there. But whether it is normal or how it will be there. Suppose here it is a normal like this you take. Then the daughter should be what happens carriers. So that 100% what would be there the female heterozygous. How you have to see here the chart it will be there. Here it is a affected. Affected means what would be affected like this you take XY. Here affected male. But it is a normal data. Normal data two configurations are possible. Like this is there, like this is there. Suppose you take like this. How it will be there like this here? Then normal data will come. There is a choice what is this normal data. But in this case, what is the choice is there? Normal daughter is possible, affected daughter is possible. Whenever it is like this cross is there, affected daughter, like this cross is there, normal daughter. But according to this conclusion, there is a scope to have affected daughters, normal daughters. But in this case, the parents, what is the parents here? The female should be a heterozygous condition. Otherwise, without heterozygous condition, what would be there? No choice to have this here, this affection in the female. But as per pedigree analysis, according to genotypes, already given in previous question, as per this, what is the conclusion we have drawn here? The female parent should be a heterozygous. But maybe choice, normal daughter is also, suppose one more daughter is there. There is a choice to have a normal daughter. Henceforth, it is what it is here. Could not have normal daughter, wrong. It is not colorblind and colorblind. Male parent is a homozygous. Male parent is heterozygous only, not homozygous. Henceforth, here what is the conclusion? Female is heterozygous. Then only there is possibility of having these daughters with affected and non-affected. So what is the conclusion we draw? Female parent is the heterozygous. That is the answer. Next year, category 37, 38 question. A cross in which an organism showing dominant phenotype is crossed with recessive parent in order to know genotype. I'll repeated question. What is the cross required for knowing of genotype? Which cross is required here? Which cross is required to know genotype? Here, what cross is required to know genotype here? Test cross. Option A, this is a test cross is correct answer. Which cross is required for the knowing of genotype? Test cross. Their option A, monohybrid, B, back cross, C, test cross, D, dihybrid cross. So what is the cross required for find out the genotype here? Test cross. So answer is the test cross for this. This question already came. Next one here, anti-rhinum, 39 here. Anti-rhinum means incomplete dominance. 39. What is the 39? Anti-rhinum. Anti-rhinum is there. Anti-rhinum is example for incomplete dominance. Two plants with pink flowers were hybridized. The F1 plants produce red, pink, and white flower in the proportion of red, pink, white 1:2:1. What would be the genotype of the two plants used for hybridization? Red flower color is determined by RR, white R smaller. Here what is the question? In anti-rhinum, red flowers are there. Red flowers, how they are indicated? Capital R. White flowers, how they are indicated? They are indicated with genotype smaller. This is the genotype of red flower, genotype of white flower. Red flower genotype capital R, white flower genotype, small r, the dominant allele, two are the recessive allele. But pink, what about the pink here? Pink is the capital R, small r. So what is the cross has been made here? This is a cross has been made. This is a pink into pink. In given question, here a cross is in antirrhinum. A cross has been made between pink into pink. This is the what is the flower? Pink flower. What is the flower? Pink flower class has been made. So find out this here. What proportion plants will come? These are the here capital R, small r gametes will come. It is the gametes. Then here capital R. What is this plant? Red. Capital R, small r, pink. Capital R, small r, pink. Then here small r, white. What is the proportion came in the given question? Red, pink, white, 1:2:1. This total information given in question only. Here a cross is made between pink flowers, in which here 1:2:1 red flowers, pink flowers, white flowers came. But what is the genotype of parents? Parent genotype, what is this here? Capital R, small r. Here, what is the actions they given? The actions, first action, here R, 4 R's given. Next here, capital R is given. Option C, what has been given? Capital R, small r given. Option D, what has been given? Small r. This is the genotype of parents. Capital R, small r. The question is in antirrhinum, cross is made between two pink flowers, which results red to pink, white, 1:2:1 has come. What would be the genotype of parents? The genotype is the heterozygous. What is this here? Genotype. Capital R, small r, heterozygous genotype. In this antirrhinum, heterozygous revealing intermediate color because here incomplete dominance. This question is relevant with the incomplete. Wherever antirrhinum, Andalusian, and snapdragon plants are given examples. So here it is the what is the case here? Incomplete dominance questions here. And select the incorrect statement from the following. Galactosemia is inborn error metabolism, and small population size result in random genetic drift in population. Baldness is sex limited trait. Baldness sex limited or sex influenced? Sex influenced. Baldness occurs in both male parent occur, female parent occur. Both baldness influenced trait. Sex limited means it confined to anyone. That is the sex limited. The sex limited means what is the sex limited? The character should be occurs in any one individual, that can be called sex limited. Suppose a hen is there. Hen is there, next what would be there here? Cock is there. In cock, fowl is there. On the head, fowl. But that fowl is not there in hen. Hen means female. Here cock means male. In male, what would be there? Fowl is there. That fowl is not there in hen. That is sex limited. That character should be limited to anyone limited. Baldness applicable for female and male. But here where influenced? Male influenced. But this option, what is the C option is correct. 14th question, C option is the correct. Here 41, sickle cell anemia. What is sickle cell anemia? Caused by substitution of valine by glutamic acid in beta globin chain of hemoglobin. Glutamic acid is replaced by valine. Valine is not by glutamic acid. But how this sickle cell anemia is caused? Change of one nucleated base pairs. One nucleated base pair has been changed, which results what happens here? The sickle shape hemoglobin turned to the what is the hemoglobin shape here? This is a hemoglobin shape. Here this is the RBC. In this here it is the RBC cell, hemoglobin is there. This is the RBC. This RBC, how it will be turned here? Sickle shape. This shape, what is this? Sickle shape. Generally, GAG is replaced by GTG. That means A is replaced by T. The one nucleated base pair has been changing. By the changing of one nucleated base pair, this what is this here? Biconcave or rounded shape, or this is turned to sickle shape. That can be called as the sickle cell anemia. You have to check your served papers here. Incorrect statement. Sickle cell anemia is substitution of valine by glutamic acid. No, glutamic. Yes, one nucleated base pair is changing. Let's see what is this changing here like this. This is the normal DNA. Here GDG is there. CAC. Here what is changing? This is changing here. In the DNA, what is the DNA? This is a DNA. Sickle cell anemia. TA pairs are replaced by AT pairs. In place of T, what came A? In place of what is the TA? T came. TA is replaced by AT. So what is the codon here? You see this here, GTG, CAC. Suppose here like this is there. What is the codon here? U is there. T means here A. What is this here? C. UAC codon. C means here G. A means here U. GUC. The codon is GUC codon. Here the codon, what is the codon? GUC. Here take 3 to 5 prime, 5 to 3 prime. Here 3 to 5 prime, 5 to 3 prime DNA strand. But in this strand you see CAC. CAC can form codon GUC. But GUC is threonine, proline, glutamic acid, here glutamic acid. First position, second position, third position, fourth position, fifth position, sixth position. Here like this is there, glutamic acid is sixth position. But here now sixth position glutamic acid is there. What is the shape of this here? RBC round shape. Here glutamic acid is replaced by valine. GAG is there. Now what would be there? Valine, histidine. Here leucine, threonine, proline. Here next in place of what is this here? It is normal shape of RBC round shape. Sixth position glutamic acid is there. Glutamic acid is replaced by valine. Here glutamic acid is replaced by valine. But how it is replacing? Change in one nucleotide change into AT pair. Then the codon in place of GUC, GAG come. But GAG is responsible for valine. So in place of glutamic acid, valine has come. Then round RBCs come to sickle shape RBCs. That cause the sickle cell anemia. Therefore, what is the sickle cell? Caused by the change in single base pair of DNA. Correct. So answer B. And here adenosine deaminase deficiency may be cured permanently. Generally, whenever permanently it will be cured, if the ADA is administration of ADA gene, option A, administrating ADA gene activators, or introduction of bone marrow cell producing ADA gene at early embryonic stage, enzyme replacement therapy, periodic infusion of dentically engineered lymphocytes having functional ADA cDNA. So you see what is the permanent cure? ADA problem is there. Anyone they have this ADA gene problem. ADA enzyme problem happened. What would be the problem? ADA gene is problem. Then ADA enzyme is problem. ADA enzyme is the problem. Next, what is the problem? Lymphocytes cannot be produced. Then immunity problems will come. But how it will be rectifying? We are going to hospital, replacing this. What would be their lymphocytes? But these frequent introduction of genetically engineered lymphocyte, this is complicated for us. But what is the permanent cure? Embryonic stage. Bone marrow cell, genetically modified bone marrow cell, introduced at early embryonic stage, it would be permanent cure. Therefore, what would be the answer? 42, B answer. For rectification of permanent cure of ADA problem, here genetically engineered bone marrow cell could be introduced at early embryonic stage is the permanent cure for this year ADA problems. The most popularly known blood grouping is the ABO grouping, because O in refers to having. What is the O? Generally IA. We are telling in ABO blood group. What is the question? ABO blood group. Here it is called as A blood group. This is called as a B blood group. But why this is called as a zero blood group? Because no antigens. Why this is called A? Its RBC, A antigen is there, it is called A group. And this is called as B antigen is there, B group. Why this is O group? O means here zero, no antigen. Therefore, here what would be there? This is called as the O group. Why we are using ABO means why AB0? Why this is AB0 blood group? No antigen on the surface of RBCs of O group people. Therefore, we are using the zero symbol instead of C. Henceforth, what is the answer? no antigen A and B on RBCs. Here what would be there? A and B. No antigens. A and B antigens are not there. No A and B antigens on RBCs. There is no A antigen, B antigen. Suppose A group people, A antigen, B group people, B antigen, AB group people, AB antigen. O group people, no antigen. So AB antigens could not occur on O group. Therefore, what is the answer here? No antigens A and B and RBC. The answer is what is the answer here? C answer. And this is already, we gave pedigree chart here, last one. This is the autosomal recessive disorder. Pedigree chart here, previously came same chart here, pedigree analysis. Now you see this here, 45 question. Which of the following condition in human is correctly matched with its chromosomal abnormality? Here, erythroblastophidolosis, 45 question. Question number 45. You have to see which of the following condition in human is correctly matched with its chromosomal abnormality. Erythroblastophidolosis means what is this Rh factor? Here, Rh factor is causing the erythroblastophidolosis here. Rh positive man, Rh negative woman, they are married. If the growing fetus is Rh positive, at that case, second birth is caused by the erythroblastophidolosis, so that it is not X-linked. Down syndrome, 44 autosomes plus X0. What is the Down syndrome here? Option B, Down syndrome. Down syndrome means 47 chromosomes. 21st chromosome is, what is the 21st chromosome is added with extra chromosome? Down syndrome, 47 chromosome. 45 plus X0, 44 plus X0, what is this here? Turner syndrome. Klinefelter syndrome, 44 plus XXY. You see the options, correct option we need erythroblastophidolosis. Erythroblastophidolosis. This is X-linked, not X-linked, wrong. Option B. What is the option B? Here 44 autosomes plus X0. This is the Downs. It is not Downs, it is Turner syndrome. Option C. What is the option C? 44 autosomes plus XXY. This is called Klinefelter syndrome. Here, what is this here? Klinefelter. This is Klinefelter. Yes. One extra X chromosome is added. Klinefelter. So that it is a C answer is correct. 45 color blindness. They are given color blindness. Option D, color blindness. What they are given color blindness? Y-linked. But it is not Y-linked. What is this here? X-linked. Color blindness is the X-linked property. So you see erythroblastophidolosis. This is relevant with what is this here? Rh factor. So that it is not X-linked. Find out correct statement from following. What is this? Erythroblastophidolosis is a Rh factor. Not having this X-linked, so wrong. 44 plus X0, not a Down syndrome. It is wrong. What is this here? Turner syndrome. This is a Turner is correct. 44 XXY, Klinefelter correct. One extra X chromosome information remaining. What would be there? We can discuss 15 more questions tomorrow class. Afterwards here we will discuss molecular basis of inheritance. One more class we will take principles of inheritance. Then we will conclude C. Then followed by we will discuss the molecular basis of inheritance. I can share this information molecular basis. Same sheet I will discuss here. You follow that sheet, that instructions here. Then I will explain.